protein engineering to using industrial scale. As you know, the phytase is a variant of the phosphatase that is classified to the histidine phosphatases. It releases the phosphate from the some inorganic ingredients. Phytase producing organisms are a variant of the organism in the nature. Between them, fungi, yeast, and bacteria are the main producer of the phytase. And between the phytase producer, Aspergillus niger phytase, because is well characterized and its gene was determined, and because of its high catalytic efficiency, we choose the gene of the phytase from Aspergillus niger. We choose the phytase from Aspergillus niger and try to express it in yeast because of some advantage of the yeast. And after that, we improve some characteristic of the phytase. Phytase have variant application in industrial application because 50% of the soil appear to be in the form of the phytate and this need to be released. And the next application and the main application is in feed as a feed additive because a lot of industrial enzymes now added to the feed as a feed additive. In our work, we have two goals because at first stage we have to express recombinant phytase from Aspergillus niger in yeast Pichia pastoris. And then in stage 2 we use the protein engineering approaches, which the main approaches which we have used is site-directed mutagenesis to engineering the phytase to improve its character. Material method we have used in a first stage. At first we choose the phytase enzyme from NCBI. Then we have done some codon preferences optimization and asked the company to synthesize the gene. The gene was cloned in Pichia pastoris, P-Pink HC vector that we bought from in vitro gene with some character like AX parameter, alpha-matic factor signal as a signal peptide, multiple coding site, and variant part that is indicated in a page. Then we cloned the Phi-A gene, synthesized Phi-A gene in P-Pink vector, and for detection of the cloning we use two forward and reverse primers. In stage 2, we plan to manipulate the recombinant phytase. So, because the phytase that we have and the wild phytase has need to be used in the industry, and for industrial application, it should be stable against the temperature, and we should have a thermostable phytase, because during the food plating, we have a high temperature like 80 centigrade, and it should be thermostable. And the second item that we used was kinetic efficiency. If we have a thermostable phytase with high kinetic efficiency is ideal for our future and for industrial application. For mutant designing prediction, at first we have we take a look on the sequence of the phytase. We compare it with the thermostable phytases from the other fungals, like Aspergillus fumigatus and some thermostable phytases. Then we check the amino acid accessibility and interaction investigation. And finally, we control the 3D structure, and we find some stabilization amino acid. For structural studies, we use the Yasara software. We use the PDB file from the website, and we analyze the structure of the phytase with Yasara. and use DSSP and what if databases for secondary structural assignments and solved exposure of the protein, and some other items. We have aligned our sequence with thermostable phytases, and we find four main points that is indicated with green, purple, yellow, and red. The yellow and the red are two sequences simultaneously. We have determined 6 points, 1, 2, and 2 point here and 2 point here. Then before doing experimental, we tried to have some simulation and modeling. So we modeled our substitutions with homology modelings, and models were validated. And then the simulation was done as a molecular dynamic that the behavior of the enzyme at 80 centigrade, that is 353 Kelvin, was determined, and after that the total H bond and the total salt bridge and the long distance hydrogen bond and H bond energies was calculated. Finally, we have selected 6 points, and the 6 point was confirmed with a simulation and modeling system, and then we start experimental mutation substitution. For experimental mutation substitution, between the variance model, we used site-directed mutagenesis. We use quick change site-directed mutagenesis kit. We have used 14 variants of primers for designing variant kind of mutation. This is the table that indicate from phytase P8 to phytase P14, from single mutation, double mutation, triple to 6 mutation, and the result. The gene was selected and was synthesized, and also the sequence was optimized, that it is accession number is in NCBI. The gene was digested and cloned in the P-pink vector, and the cloning was confirmed by colony PCR. Then the recombinant vector is transferred to the Pichia-Pasteur's host by electroporation systems, and the transferred recombinant systems was controlled on the pad media. Then integration of the gene into the whole genome is detected, is confirmed. The genome is extracted, and with a specific primer is confirmed. Then recombinant Pichia-Pasteur's with integrated phytase in the genome was expressed at first in BMGY medium and then in BMY medium. The expressed protein was determined here with phytase activity of 179 unit per ml and a specific activity of 66.7 unit per milligram. Then it is purified with a FPLC method, and you can see the smear of the protein that is related to hyperglycosylation. Then 2 steps of purification was done, and the specific activity is increased from 66 to 89 and then to 121. As I told, the smear was the reason of the smear was the glycosylation. So we use EndoH for deglycosylation, that you can find one smear is changed to the 1 bond. Then the kinetic parameters of recombinant protein is determined, like KM, Vmax, KCAT. And the recombinant phytase temperature graph, pH graph, was determined. It is determined that the enzyme has the best activity at 60 centigrade, and the pH of have two optimum pH, especially in 5.5. Phytase thermostability is determined, recombinant phytase. And then we have done the same experiment for engineered phytases. As I told, 6 point was determined for point mutation. The 3D structure of the phytase area, the 3D structure of candidate area is determined, and the 3D structure is, you can see, it is A is wild type, B with 3 mutations, C with all mutation is determined. The quick change site-directed mutagenesis as experimental work has been done, and you can see the all bands. And the confirmation of the mutation was done by sequencing method. Then the substituted point was again cloned and expressed in Pichia-Pasteur systems, again in a PPEG system, and then transferred to the Pichia-Pasteur by electroporation system, and a screening and expression was done. Mutant phytase thermostability analysis shows that among 14 mutated items, single mutation hasn't a significant variation. You can see the wild type and the single mutant hasn't significant variation. But among the double mutants, we have an improvement of 14 and 15 in P7 with these 2 point mutation. We have 14% improvement in P8, 15%, and P10 we have 12% improvement at 90 centigrade. Between the triple mutants P9 and P11, also we have improvement at 80 centigrade, that is feed plating temperature. In P9 we have 24% improvement, and in P11 we have 8.5% improvement. And also we have mutants with 5. We have a mutant with 5 point mutation. And between the 14 mutants, P9 and P12 were the best items, that shows 24 and 22.6 respectively improvement at 80 centigrade. That could be a good candidate for the industrial application. And also P13 and P14 with 6 P13 with 6 substitution has a higher thermostability in compare with the wild type at 70, 80, and 90 centigrade. That could be good ideal systems for industrial application. About the kinetic efficiency and the kinetic of the mutants, between the mutants, these 4 mutants shows the improvement in kinetic efficiency. That 4 items have better thermostability and better kinetic efficiency. As a result, we can say that between the 14 mutant P13 and P14 shows 31 and 6.5 respectively higher catalytic efficiency compared with wild type, and the P9 and P12 shows 35 and 25% higher catalytic efficiency. might be substitution of the this substitution at this area might be because of the arginine is has a positive charge and might be have a contact with the phytate because of negative charge, and could be effect of the could be effect on the interaction. Analysis of the, because we have experimental we have done the molecular dynamic with simulation and modeling. We compare our experimental result with the molecular dynamic result. We have these 4 items of mutation with the best thermostability and kinetic efficiency. That the result of the molecular dynamic also shows that in a wild type, in compare with a higher thermostable mutant, in higher thermostable mutant we have higher H bonding. That the higher H bonding means that higher thermostability, and also about a strong hydrogen bond. We see the improvement in the in a mutant in compare with the wild type. H bonding pattern and also H bonding energy changes. We have seen that in next slide. I will show you. Also, we compare the wild type and the mutants, and at 0.205, that we see that we have C extra glycosylation site. That the higher glycosylation means higher thermostability, and also in double mutated points, we see that the pattern of H banding is changed, and also extra H banding seen. Also, in compare of the wild type with the mutant, we see that the distance also is changed. In mutant, we have a lower distance that and higher in H energy, that it could be a result to the higher thermostability. At end, thanks for your patience. Any question? Yes.
Thank you for the presentation.
Thank
you. In your research, point mutation improves the thermostability of phytase.
Yeah.
So it is possible to combine point mutation, for instance, P12 with P14.
Yeah, it is possible. We are doing. It might have positive effect, but before it, we will do again molecular dynamic, and if we have positive molecular dynamic result, then we will do experimental result. Yes, might be, yes, because we have combination of because the improved phytase has a combination of the single mutants. Might be I have seen some research that 14 point mutation together have a better result. I'm not sure. We are trying to do in next steps. Thank you.
Yes. I just have one question. So what's the prevalence of use of phytase in the feed industry?
We add the phytase to the animal feeds to release the phosphate in inorganic compounds, because you know that in a wheat in a feed ingredients the phosphate is not released, and also in a monogastric animals, because it didn't release the phosphate is not released. So adding the phytase to the feed animals, it can help to release the phosphate that can have a positive effect on animals. And also another alternative way is to add a chemical phosphate to animal feed. But if we add any chemical phosphate, it will go to the water and have some pollution, environmental pollution. So it is a lot of commercial companies that are producing phytase as a feed additive, especially in feed.
So almost 100% of feed they have a phytase in the formulated feed.
Yeah, most of them, yes.
Most of them.
Yes, most of them. Yes.
Yeah, thank you. Good presentation. I saw that you checked the effect of pH and even temperature on the activity of the phytase. I don't know if you did test the effect of these factors on the KM.
On the GAN,
On the KM of the enzyme.
we have checked the thermostability and kinetic efficiency. These two factors between the wild type and mutated. We don't check the pH yet, but we should check it too, because in the mutant and the wild type there wasn't high variation between the wild type and the mutant. But we have checked the kinetic efficiency and also thermostability.
Okay, because when this activity are there, it may not actually translate to affinity for the phosphates. And that's what I'm just talking about.
As I get you, it means that when we have more thermostability, we have lower kinetic efficiency. Yeah? I got you correct? Yes, you say correct, but it depends on the structure of the enzymes. If the enzymes go tighter, the thermostability will go higher, but the kinetic efficiency come down. But I don't know why. And also in a lot of research, we have seen such data that the thermostability and kinetic efficiency can improve simultaneously. Might be the reason is that the conformation of the enzyme doesn't change highly. We can check it with some other experimentals like TM, melting temperature. We can calculate the TM, and after that we can understand that the enzymes goes tighten, or might be the flexibility of enzyme also improve, and some, for example, inner enzymes come outer or some variation. I don't know. We can check it in future. Thank you.
Just comment, and you don't have to answer. Thank you for your good presentation.
Thank you so
And
much.
the phytase is very important feed additive, so it can improve the quality of the excrete.
Yeah.
And how maybe in the future you need to register your GMO approved by your government.
We have some GMO regulation at first. Now you're working on fermentation systems, and also we are trying to do some criteria that IVO asked us about the GMO application of the phytase. But I think if we pay attention to criteria, because a lot of commercial phytase are using now. A lot of companies, as Dr. told, 100% of the feeds are with phytase. Our country also in Iran, we have a specific regulation for GMO application. We have the list of this criteria, and we are doing pay attention to such things. But before that, in parallel, we are working on fermentation systems, higher fermentation systems, and also I told that we express the phytase in Pichia pastoris as a host. Also in parallel, we express the Pichia pastoris in cellular polymorphize and other yeast systems for industrial application. For GMO regulation, also we have a collaboration with the German companies that they can help us about ISO and some items. I don't know, I just know the scientific. Thank you so much. Thank you.